Hi everyone. I think since the gong just started, it's time to start. Thank you so much everybody for being here for this session. We're really excited for the speakers that we have lined up. And I'm not going to take more time from Emily Meineke and just give her the stage.
Hi, everybody. Thank you for coming. I want to thank the hosts for inviting me and for putting together this symposium. After looking at the schedule, I got really excited that a lot of you are early career and that you're starting to use these specimens for creative purposes. And I hope to talk about that and that keeps going. I'm also thankful to be here because I have a 1-year-old at home, and last night was the first night that I've slept through the night in about a year. So I thought some of you could relate to that. So I'm going to start by talking about some work that I did during my postdoc, and then I'm going to fast forward to some work that's been done in my lab at UC Davis in recent years. So during my postdoc in March of 2018, I was in Japan to visit the National Herbarium for research, and my trip was not supposed to overlap with the big cherry blossom bloom that happens in Japan, but it did, and the reason was it was between 5 and 10 days earlier than average. And in fact, 2018 at that time, the cherry blossoms happened the earliest they have in history. So the reason why we know this about the cherry blossoms is that people have been keeping track of when they bloom for something like 12,000 years. So what you're looking at right here is time on the horizontal axis, and then on the vertical axis is the date of cherry blossom peak bloom. So the dots that are lower means that in a particular year, those cherry trees bloomed early. So if you're looking here at 2018, it's some of the lowest points for most of the cherry blossom trees. So this is sort of a general phenomenon among a lot of plants that they are flowering earlier and leafing out earlier than they ever have. And there's been some concern that this is going to lead to phenological asynchronies, phenological mismatches between plants and the insects that pollinate them and eat them, and in general interact with them. And while we have data on plants, some select plants, and how they've responded over the period of recent history to human-driven change, we don't have comparable data on the species that they've interacted with. And so you can imagine you can't go back in history and figure out what microbes were associated with these trees and what insects were eating them, what insects that were pollinating them, et cetera. So I'm going to talk about a specific kind of species interaction today that's very consequential ecologically and also is not very well represented in historical records. So what I'm focusing on are insect herbivores. So insects have been eating plants for something like 400 million years. And in the past 12,000 years or so, we have disrupted these relationships and changed them. And historically, the interactions between insects and the plants that they eat, the coevolution that they experience has led to most of terrestrial biodiversity. So you can imagine that these human-driven changes to the environment potentially could have big effects on ecology. So these are birch sawfly larvae, and I just wanted to illustrate the big impact that insect herbivory can have in really small amounts. So the birch sawfly larvae, when they remove something like 3% of leaf tissue, they result in 20% less sapling growth in terms of height. So insect herbivores can have really big ecosystem level effects. So on the one hand, there are some pretty clear predictions about how insects should respond to climate warming. So what you have here on the horizontal axis is temperature, and then on the vertical axis you have some metric of insect performance, think like the number of eggs produced or how well they survive. And these metrics go up until you hit T optimum, and then they rapidly decline as temperature goes up to the critical thermal maximum. So theory predicts that as long as herbivores are below their thermal optimum initially, climate warming should benefit them in terms of their fitness. And there's some evidence of this from the paleo record. So these are each photos of a different type of insect herbivory on a fossilized leaf. And during the Paleocene-Eocene thermal maximum, when temperatures were raised something like 5 degrees centigrade over 10,000 years, both the abundance and the diversity of insect herbivory went up. So we have some evidence that in practice, the insect herbivory metrics that we would look at today went up in the fossil record. The problem is that actually the Paleocene-Eocene thermal maximum, that temperature rise happened quite slowly compared to climate change today, so it's not a direct analog. The other thing is, and many of you have probably heard of this and similar articles showing across studies that in a lot of cases, insect biomass and abundance is actually going down, which is counter to the story on climate change. And so this is a figure from the most recent, I would say, and comprehensive meta-analysis on this topic, and those brown bars show that terrestrial insects are in decline. Okay. So I started seeing all of these studies and started wondering, like, well, what does this mean for what we know about actual interactions between insects and plants? So I looked at studies that track insect herbivory or insect damage to plants, and I said, well, maybe I'll reconstruct that over time using these studies. But it turns out that most of these studies, according to this histogram, only last 1 to 2 years, probably because of grant cycles and various things. And so there really isn't a lot of long-term data tracking insect herbivory. Okay, so enter herbarium specimens. Herbarium specimens have been variously collected since the 1700s or something. And I think when I initially started this project, I thought that they wouldn't display many signs of insect damage because people would be biased against collecting damaged leaf tissue. But it turns out if that's what they're up to, they're bad at it because it shows up all the time. So this is the oldest specimen at the Carnegie Museum of Natural History, and it has little insect chewing holes in it. So I started to notice that sort of generalized chewing that you can't attribute to any particular insect group, but also really specialized insect feeding. So these are megachilid bees that make these really characteristic 3/4 moon-shaped holes in leaves, and then they take those back to their nest and they create a little nest hole for their young. And this is that same type of damage on a herbarium specimen of blueberry. We can also look at really specialized damage. So this is a leaf mine on the left that's created by the moth species on the right, and you can actually use the frass or poop patterns within the mine to determine what insect made the damage. So we can actually use herbarium specimens to reconstruct insect diversity when we don't have those data. So I got really excited about this as a data source, and I think the capacity for this it's way beyond me for sure. So each of these dots is an herbarium, and the dots that are in white are herbaria that have more than 1 million specimens. So there are over 400 million herbarium specimens that have been documented to date. So knowing this, and knowing that I really wanted to understand the history of herbivory recently, I had to figure out a place to start. So I got a postdoc at the Harvard University Herbaria, and I decided to focus my studies in New England. And I did this for a couple of reasons. One is that Harvard has a really good collection of New England plants. The other is that New England has warmed a lot over the winter. And historically, presumably because New England is so cold, it has killed off a lot of insects in the winter time. So what this map shows is that since 1970 in the deep red, that New England has gone up in temperature during the winter by over 1 degree Fahrenheit. Sorry, it's in Fahrenheit. Blame NOAA. So my prediction, again, was that herbivory in New England has increased over the past century as temperatures have gone up and been more favorable for insects that eat plants. So then I had to figure out which plants to focus on, and New England has a really diverse flora. And the 4 plants I decided to focus on, they have important ecosystem functions, they support rare insects, etc. So Desmodium canadense supports rare insects, Vaccinium angustifolium, blueberries, which we eat and animals eat, and then Quercus bicolor and Carya ovata also produce nuts that insects and various other creatures use. So I wanted to make sure that if I was going to focus in on a few plants, that they were important plants for the forest. So then I ran into this issue of people collecting herbarium specimens to document floras and not for me to quantify herbivory, which I had a little hissy fit about, and then I decided to get to work and figure out how to quantify herbivory on the specimens like an ecologist, and got this grid-based system up and running with some PVC pipe and some string. And these boxes are labeled from 1 to 40. And so I chose randomly, using a random number generator, 5 boxes that had enough leaf material. And I marked them as either yes, an insect had chewed them, or no, an insect had not chewed them. So what I mean by that is this sort of damage, so insects that are chewing on either the edge of the leaf or the interior of the leaf, because it was by far the most common type of insect damage that I saw in specimens. So this is what that looks like on a whole specimen, zoomed in here. What you can see is that an insect has eaten through a lot of this plant. And if you kind of squint, you can see what we call a reaction rim. So I wonder if I have a pointer. Do I have a pointer? Okay, nope. So there's little sort of brown line around the insect damage that I've blown up, and that's the plant reacting to the insect damage, saying like, I'm gonna pump this full of defensive compounds and also seal this up so that the insects can't do their damage. So that differs from insect damage that happens inside the museum, which was something I had to contend with. And insect damage that happens inside the museum more often, for instance, happens along a leaf at multiple layers. So you can see that this is damage probably by a tobacco beetle that made its way along the leaf and also ate the leaf margin, which is unusual in the field. So I got pretty good at telling the difference between these two. And now I'm going to go directly to the modeling and the results. So the response variable here, grid cells out of 5 with insect chewing damage, and predictors of interest were either year or winter temperature in 2 separate models because they're going to be correlated with each other. And then we used latitude, longitude, day of year collected, and plant species as covariates. Okay. So this is the first result. On the horizontal axis is year that an herbarium specimen was collected. The probability of herbivory showing up on an individual specimen is on the vertical axis. Each dot is an herbarium specimen. Herbarium specimens that had some sort of herbivory in those 5 boxes are on the top. Ones that didn't are on the bottom. And as you can see, with each one of these species, herbivory has gone up over the past something like 112 years. Okay, so now let's look at winter temperature. So if we look at winter temperature on the horizontal axis instead of year, 3 out of 4 species behave as we would expect if the hypothesis that we had at the beginning was correct. The one that doesn't is Quercus bicolor. I've always said during these talk, like if someone can figure out what's going on here, please tell me, but I haven't gotten any clear answers yet. Okay, so wrapping up this part of the talk, herbivory is increasing in New England. And this might be because herbivore ranges are expanding. We have some evidence from that from our paper as well that I don't have time to show. Or that native or non-native insects that are residents in New England are surviving better or eating more because it's warmer. So the focal plants in this study provide important ecosystem services that may or may not be affected by these changes in herbivory. I think it would be cool to follow that up. And then I think that herbarium specimens offer this opportunity to study herbivory and insect diversity more broadly. But then when I got to UC Davis, for multiple reasons, I sort of decided, well, let's pump the brakes and see what these herbarium specimens are actually good for and see what they're representing accurately and what they aren't. So do herbarium specimens provide biased data? Probably. People are not going out in the field and saying, let me get a medium damage specimen from each location. They're saying, let me get the nicest specimen, or I don't care, I'm cold, or whatever. So they're probably biased. And it might be that these down biases cause us to see things in the data that aren't real. And I'm going to talk about that in just a second. So I wanted to quantify biases that originate from collectors and curators. And so one question is, does herbivory collection and curation lead to down biases in herbivory on specimens compared to random samples of whole plants? And then another question that's a little bit more complex to explain is, do these down biases, if they exist, swamp out real differences in herbivory across taxa? What I mean by that is, if you look at this hypothetical graph with species 1, species 2 on the horizontal axis and some metric of herbivory on the vertical axis, in this hypothetical scenario, herbarium specimens in the open circles have lower herbivory on average than ecological random sampling, but they track each other, right? They're highly correlated. However, if herbivory is so low on the herbarium specimens, it might be that they actually don't represent differences among, for instance, plant species. So we wanted to test this hypothesis, and we used the study site, UC Davis Arboretum, because it was COVID, and we needed to do something that was very local. It ended up being a really cool study site for us. We chose 14 species growing in the Arboretum that had a number of individuals we could work with, but also had insect damage. So a variety of native trees and shrubs, which all have herbivory. And this is what we ended up with, something like 452 plants spread across the UC Davis Arboretum. Okay, so the first thing we did was we pre-surveyed focal plants for insect herbivory. So Ian Pierce came up with this method that's very silly, but works. So you put your hand to your nose like this and you spin around, and then you go like this, and the leaf that's closest to you is the one that you assess for herbivory, or the branch that's closest to your finger. So we did this for 20 leaves per tree on each cardinal direction per plant. And we quickly realized that, like New England, the most common type of herbivory in our part of California is chewing, so we focused on that. And then we recruited volunteers from throughout the community to collect herbarium specimens for us. And so the volunteers were trained using a standardized protocol by our herbarium director, and they weren't told what the real purpose of the study was. They were told that they were there to help document the Arboretum and learn how to press herbarium specimens. And this was actually, it was such a fun way to engage during COVID with the community. So this is our technician, Dave Eng. He's very happy with his load of herbarium specimens that he's collected. And this is him assessing herbivory on the leaves of the specimens. For this project, instead of using the grid-based method, we estimated % leaf removed per leaf. Okay, so this is the first result. What you're looking at in blue is herbivory from specimens. What you're looking at in green is random estimates from whole plants. And the proportion of leaf area removed or chewing is what's on the vertical axis. So overwhelmingly, you can see that herbivory on whole plants is a lot higher, right? It's on average 3 times higher than herbivory that's estimated on specimens themselves. But, and for some reason, Windows has ruined my beautiful error bars here, but they're small. So this is a Bayesian model that looks at the concordance of herbivory between species that were assessed from herbarium specimens or whole plants. So each dot is a species. And what we can see here is that actually herbivory, even though it's lower on the herbarium specimens, if you look at the axis, actually it tracks quite well, and that species with more herbivory in the field also have more herbivory in the specimens. And this explains something like 46% of the variation in the model. Okay, so that's collector biases and how those might affect the estimates of species interactions from specimens. We also wanted to look at effects of curators. So we had several curators take our specimens and we said, curate these how you would. A lot of times curators will cut off leaf material if people have collected too much, or they'll arrange them in a different way. And so we then assessed what herbivory on the curated material was versus the material that the curators threw away and put on a different sheet and behind that. And what we found was that actually our curators really didn't make much of a difference in the estimates of proportion leaf area removed on the specimens. So putting all of that together, herbivory cached in herbarium specimens is definitely down biased. And fortunately, herbivory on the specimens also mirrors herbivory on whole plants assessed randomly. So we do think that herbarium specimens can be accurately used to assess differences in herbivory among plant taxa. So while collectors are associated with down biases, either because they're systematically going to go in the field and collect like undamaged branches, or because herbivory is clustered, and if you go out and collect one branch, you're more likely to get a 0. They are down biased, the curators were not. So perhaps they're more occupied with other features of the specimens. Okay. So our future directions actually overlap with evolution, which is great because I've talked about ecology so far. So a PhD student in my lab, Grace Horn, is really interested in looking to plants that have insects that have declined due to broader insect decline, and to see how the traits on those plants may or may not have changed as the herbivores have been lost. And so if there is this massive insect decline happening, which for the record I do think that there is, it should have evolutionary impacts on plants that are trying to otherwise defend themselves against these insects. So we'll be interested to see how that shakes out. Additionally, Lauren Azevedo-Schmidt is looking at specimens across the US, maybe across the world, and looking at different regions within the US that have warmed and changed in climate differently to see if that has affected how herbivory has changed. So for instance, in the southeast, temperature has gone up less than it has in many other areas of the US, so we're interested to see if that has affected the trajectories of insect damage on plants over time. And she's starting right now in California with this magnificent tree, the valley oak, which supports a really wide array of galls that you can see presented in the bottom right. Okay. So I have presented my methods to you in kind of painstaking detail instead of giving you a bunch of results slides today. Number one, because this work is kind of new, but number two, because I think it's so useful to see how someone has gone through various challenges associated with deriving good data from specimens. I know that a lot of my cohort in the PRFB, there was a bunch of people who are working with herbarium specimens and various other types of specimens. We spent most of our tenure as NSF scholars trying to figure out how in the hell to get our data. And that really inhibited our progress. So if anybody wants advice or wants to just chat about how annoying that is, I'm here for you. And I think that there's a lot of potential here on this map for deriving all kinds of interactions-based data, not just herbivory, but pollination, et cetera. So again, I'm so excited that there's so many early career folks here who are pursuing that. Okay, lots of people make this work happen, especially the volunteers in the Arboretum and people in my lab who came in during COVID. So I'm thankful to them that they masked up and came in, and our herbarium staff and volunteers also helped us out a lot. So thank you very much. I'm happy to take any questions that you have.
I'm curious what you think about like you showed the overall declines in insects but then you have the increases in delivery. What's your thought on reconciling?
Yeah, those are seemingly incongruous, aren't they? What I think is happening is I think there are a select small group of herbivores that are really benefiting from climate warming and that are just going gangbusters on these plants. And I think they're generalized herbivores, which is why it shows up on all 4 plant species. A lot of the herbivory that I saw looked very similar. So there's a new name, the spongy moth. I don't know if, you guys are familiar with that one. It eats a lot of these plants, so it could be the culprit here. I don't think it's that a huge diversity of herbivores are benefiting from climate warming. Yeah.
I wondered from seeing your data if there wasn't something going on with evolution of resistance.
Hmm.
Maybe with predators.
Evolution of resistance.
Yeah, so how would resistance track? So you're basically increasing your delivery, and then the Quercus like had the downslope. So I don't know, maybe it evolved resistance to the delivery over time. And then
Yeah.
my other thought was maybe you have an increase in herbivory in those 3 species because increased temperatures are just knocking down herbivory resistance pathways.
Yeah, that would be so interesting. I've wanted to look at the chemical composition of those leaves. That's such a good idea. Yeah. Anybody else? Yeah, Rob.
Hey, Emily, I was just thinking about the mention of the spongy moth. That's an outbreak species, I think, right?
Mm-hmm.
I'm curious, can you detect these sort of cyclicities and outbreaks from these data? And how much does that play into kind of picking up that signal of change?
Yeah, I've thought about this quite a bit. I think that herbarium specimens are best used for species that don't outbreak but are more consistent year to year, or at least have smaller oscillations, because I think that collectors can avoid, entire trees that have been defoliated. But I think if the defoliation is low enough, you could probably detect some of this. Yeah. Does that mean 2 minutes? I think that's what it means. Okay. Okay, thanks everybody. Oh yeah, go ahead, Oliver. Yeah.
You showed there were these dots like showing the temporal distribution of your data for the first study that you presented.
Mm-hmm.
And I think it's a general pattern that there's much less connected in the last years.
Oh, so fewer specimens?
So I think that's a problem for this time series that we want to continue with, right? Is this what you think about this?
It's not my favorite. Especially with what happened to the Duke herbarium recently, I'm also concerned that the downfall in collecting is associated with just a lack of funding for these institutions that are accessioning these specimens. I think that not every collection has this problem. So the Harvard University herbaria especially is like starting to max out, and so I don't think there have been as many people collecting. But for instance, I went to Japan, and just, citizens of the local region will accession specimens there. And so there were all of these specimens from the 90s. It was amazing. So, and recently. So the distribution of times was more even there. So I think there's room to select particular collections for their benefits. Yeah.
That was a great talk, Emily. I got one question that's related to curators. Do you see, if you take a specific herbarium and you look at it over the last, say, 200 years, do you see curator bias in the quality of the leaves in terms of some curators were more likely to take those pristine specimens versus the next curator came along and see like a bias as you go along?
So usually the curated material is thrown away. So we can't look at curated versus not over time, which is, not my favorite either. Yeah, it's the source of uncertainty for sure. Yeah.
Kind of related, have you considered repeating the experiment where you compare with collected specimens to forage with like a different group of people or people who've been trained for longer or different type of curators?
Yeah, so our curators ranged from people who had never collected specimens to experts. And so we did collect a range of people. And we also had them do exit surveys telling us like what they were looking for or not looking for. And none of them really talked about herbivory. They were like, it's something that I thought about, but really I wanted to get the reproductive structures. So it seems like, at least in our region, that that's really not what people are thinking about as their first goal, which is nice for us. But yeah, it would be an interesting thing to repeat in different places to see if regionally people have different preferences. Mm-hmm. Okay, thanks everybody.
Good afternoon, everyone. My name is Qindong Tang, and I'm a postdoc working with Ben Warren in the Natural History Museum in Paris. And as we all know, that we're now facing a huge global diversity loss. And here in the map, there's the warmer color indicating the regions that have less natural diversity remains. And the question of why certain species are threatened to extinction while other species are relatively at little risk is of really important questions for our future, and yet it's poorly be understood. And it is first that some explanations for why, case-specific explanations for why there's differences in extinction risk is always more broadly statement of certain taxa or communities. And second of all, the population decline to extinction is inevitably dependent on the exposure on the environmental changes and its susceptibility to such changes. And actually, masquerading archaeology has provided us a very unique systems to investigate such factors might explain the differences in susceptibilities. And in our study, we are mainly focusing on two volcanic islands next to Madagascar. One is La Réunion and one is Mauritius. And the uniqueness about the system is there's no settled human population before 400 years in present. And also during this, since first human arrival, there's large habitat destruction on those islands. And also there's a very nice record of major environmental changes, including the habitat destruction and the main keystone species extinction, and also the introduction of exotic species. And also furthermore, we are able to collect pre-human Asian fossil samples. Plus we also have a nice collection of historical topaz samples for the last 200 years when the major environment changes happened. And then we can compare those samples with the modern samples. And in our study, we are actually comparing 4 different pairs of bird species. And the first pair of species are only on La Réunion Island, and the other 3 species pairs are on Mauritius. And for each species pairs, we are including 1 common species and 1 rare species, and both of them are closely related and also sympatric. And also 3 rare species of those birds are actually endangered species. And 1 bird species on La Réunion actually also has very limited distribution range only on the highland that we consider as relatively rare than the common ones. And for we collect both like Asian fossil samples, pre-humans, but those samples are only be available on the Mauritius Islands due to preservation conditions. And also we have 9 to 11 historical topaz samples for each species, and also we're including 9 modern blood samples. And for the Asian samples, we are doing high-res sequencing aimed at 10% of genome data. And for the historical topaz samples and modern blood samples, we're doing whole genome resequencing. And on top of that, we also obtained 8 reference genomes per species. And there are 3 major hypotheses we want to test. The first is whether the rare species already had a smaller population size before the human arrival. And the second is whether rare species has already lower diversity or higher genetic load prior to first human arrival. And the third is whether rare species show less evidence of adaptation to anthropogenic environment changes. And in this talk, I'm going to present most preliminary results, only focus on 4 Justophus species. And also there's one thing I want to emphasize is for the one rare species on Mauritius, the Mauritius olive white-eye Guralnick actually is a really critically endangered species that is only around 300 individuals left on the island, and they has really restricted distribution range, shown in the red circle within the national park only. And here is a brief summary of the current obtained data. And in the first panel, it's the sampling map. For the left one is Mauritius Island, and the right one is Réunion Island. And the different colors represent different species. The first 2 colors are the 2 species on Mauritius, and the light yellow one is the critically endangered Guralnick. And the last 2 colors represent the species on the Réunion Island, and the light yellow one is olivaceous, which is considered to be the rare species on La Réunion. And also the different shapes of dots indicate different type of samples. The circle ones are the modern samples, and the triangle ones are the topaz samples, and the subfossil samples are indicated in red square. And one thing, nice thing we have is for the historical samples of critically endangered Guralnick species, we also have the locations outside their distribution range nowadays. And also, of course, there will be more data to be added soon. And here, the first result I'm showing is the PCA using a PCA-ENST method, which is likelihood-based. And nicely, we can see first here is we have 4 really clear clusters corresponding to 4 different species, including both historical samples. And also, one thing I want to say is for the subfossil samples, we're only able to know that they belong to one of the species, but we don't know which one they belong to. And very luckily here, through the PCA, we can see at least we have 3 of the 5 subfossils are clustered nicely within Guralnick. And also, we have 1 subfossil clustered within the Mauritius. And also, there's 1 subfossil we cannot really know what's the genetic background, and we think that's due to a large missing data. And also, one interesting thing is within critically endangered Guralnick species, we also find another cluster, which only clustering the historical samples, including pre-human subfossils and topaz samples. And next, we did a mixture analysis based on the same data set. And this method is just to infer the individual ancestry. And very surprisingly, is that the best K is equals 5 in the analysis, which showing actually there's also structure within the Guralnick, which is also consistent to our previous PCA analysis, that there's another pink ancestry component that all the topaz samples and the subfossils from Guralnick are in this cluster. And also another interesting thing is if we look at K equals 4, actually this pattern still holds. And actually it's also not very surprising because due to the previous study on the systems, actually the Mauritius and Bubonics are considered to be sister species, and their divergent times is also quite recent. And then I also calculate heterozygosity and pi diversity to comparing between modern samples and the topaz samples for different species. And first, when we look at the heterozygosity, we can see that all the modern samples has lower heterozygosity comparing to topaz samples. This is quite expected. However, the critically endangered Guralnick species has the highest modern heterozygosity among all the species. And for pi diversity, it's quite surprising because it shows we have all the modern samples has showing higher pi diversity than the historical samples. And also the pi diversity is highest in the most critically endangered species, Guralnick. And for this, we think it might due to the ancestral population structure, because when the habitat has a large construction, constructed, like different population with divergent might come to a more a smaller region, and the gene flow between them might cause this pattern. And this is also quite consistent what we found in the both in PCA and the admixture analysis. And also the explanation why we found higher pi diversity in the other species is we think that if we look at the sampling map, actually all our modern samples has covered a large distribution range of the species, while the topaz samples may only restrict it to certain locations and cannot really reflect the whole ancestral genetic diversity. And furthermore, I looked the allele friction spectrum to comparing also both the modern and topaz samples. And here we can see that all the rare species show the evidence of loss of rare alleles, especially for Colonotus, that there's large reduction of the rare alleles when we're comparing modern and topaz samples. This is quite we expected and might indicating a bottleneck. And furthermore, for more detailed demographic history, I used SMC++. This method has including like multiple genomes, which is be able to have more resolutions in more recent time scale. And here we can see that for the analysis in Mauritius Islands, we can detect that there's a long-term decline found in critically endangered species Colonotus. And also very interestingly, the decline also happened in Mauritius, but it is exactly started at the 400 years before present. That is corresponding to the time that the human arrives on the island. And on the La Réunion Island, also we found that the relatively more rare species, olivivaceous, also showed less effective population size, which is also kind of fit our expectations. And furthermore, also more interestingly is that in the right part, we detect all this like population size drop in all the curves. And we think this is also very compatible for the previous study in this speciation system between those island birds that fit it might indicating their colonization event on those islands and fits the hypothesis of double archipelago colonization. And for the future work, we are now having more data coming, also including the other two species pairs. And first, we want to really like leveraging pre- and post-human historical examples to refine demographic inferences. Especially, we are going to use more like demographic modeling, also to test whether the potential influences of this ancestral population structure. And second, we want to evaluate the change of mutation load and the breeding coefficient between modern and topaz samples. And furthermore, we also want to test for signals of adaptation using a haplotype LD-based method. And I would really like thanks for my supervisor and Ben Warren, and also our collaborator Yann Bonjour, and also special thanks to Catherine and Sean. And both of them did a lot of work on generating subfossil and topaz data, and this requires a huge amount of work. And also special thanks for Julian Hume, who did the fossil identification, and all the other collaborators. And thanks for your listening. Yes, please.
Hi, really good talk. I'm kind of curious, would you expect to see any sort of different patterns, considering you're looking at species that are currently rare and thus persisted over a long period of time after human entrance to the island, compared to species that have already gone extinct here?
In our system, actually, we don't include extinct species. It's only all, they're still on the island. It's just like goes through some huge bottlenecks. That's our.
Yeah. Would you expect to see any sort of different pattern than what you are finding, though, if you were looking at species pairs where one went extinct?
I think maybe there will be some similar patterns, but I'm not sure because now I don't have the data for extinct species, so I cannot be sure. Because also I think the more specific case in Guralnick is we found this ancestral structure, which is already showing some different result you can expect for endangered species. For example, the heterozygosity loss is not large enough than we might expect it. Yeah. So.
Okay. Hi, everybody. Thank you for coming to the session, which has been really great, and my talk in particular today. My name is René, and I am currently a postdoc at Drexel University, but today I'm actually going to talk about work that I, along with many, many Filipino and US collaborators, did on a project during my PhD. And this is work on tracking Anthropocene genetic diversity loss in the marine tropics. So recent studies have estimated that between 5 to 6% and sometimes as much as 10% of genetic diversity has been lost worldwide since the onset of the Industrial Revolution, with a lot of this loss being attributed to anthropogenic pressures like overharvesting, deforestation, and habitat degradation, just to name a few. But the studies that inform these statistics are relatively limited in scope. Most are traditionally done on charismatic fauna that live in temperate latitudes like the California condor or the European brown bear, just to show a few examples. And so it's less clear whether fauna in frequently underrepresented regions of the world, like the tropics, for example, have undergone declines of a similar magnitude. But it's really important to know because these often are some of the most biodiverse and vulnerable regions in the world. And to a lesser extent, it's also somewhat unclear the degree to which marine species have experienced any declines in genetic diversity or genomic change over the past centuries as well. While some studies have highlighted genetic diversity loss in marine populations, others are a bit more inconclusive, including recent temporal work on Atlantic cod, which found no real loss in genetic diversity despite continued and heavy fishing pressure over recent decades. And so this project is trying to kind of look into some of these unknowns and tackle and ask how or whether marine genetic diversity has declined in fish populations over the past century in the Philippines. And the Philippines are a particularly interesting region to look at these questions for a few reasons. So first, it's in the Coral Triangle in the Indo-Pacific, so it's one of the most biodiverse regions in the world. It has the highest levels of marine biodiversity. And the Philippines have also experienced increased anthropogenic change over the past century, including ramping up of coastal development, ramping up of commercial and industrial fishing, like this figure shows here. And so we are, and this is kind of why I'm in this symposium, looking to ask and answer this question by using historical collections from the Smithsonian in Washington, D.C. So these are collections that came from the U.S. Albatross Research Expedition that went all over the Philippines and elsewhere in the Indo-Pacific in the early 1900s and collected thousands and thousands of fish from hundreds of locations, stored them in rum or ethanol, because that's what we had back then, and brought them back to the U.S. And so we can extract DNA from these samples and pair them with DNA from recently collected fish in the same locations and in the same species to see what's changed in this time frame. And it kind of gives us a period of before and after, before and during some heavy anthropogenic change. And so this broader project is looking at lots of different changes in lots of different species, but I'm just going to narrow in on like one small subset today. And so we're looking specifically at changes in genetic diversity in two species, two ponyfish species, Gaza minuta and Equiletes laterofenestra, both collected in Jamilo Cove, Luzon, which is really close to Manila, the capital of the Philippines. And these species share very similar life history traits, so they're both nearshore demersal species that have short generation times of about a year. And they're also, I think it's important to note, commonly targeted for fishing, so they frequently show up in markets in the region. And so I'm going to gloss through our kind of sequencing here, but we used a capture bait approach for library prep and sequencing, after which we were left with these numbers of individuals, and these individuals were genotyped using hard calls at these SNPs. And so what you're seeing here for the numbers in each time point are the number of individuals that we ended up being able to use that have actual genomic data we incorporated in our downstream analyses over the number of individuals that we initially attempted to sequence. And you can see these numbers are a little low, especially in our historical time point. And some of this is just because working with historical specimens are tricky. They have damaged reads, they have fragmented reads that can result in poor sequencing output and low read depth. And so we certainly lost individuals because of that. But we also ran into issues of cryptic species here, hidden species diversity that we really weren't expecting. And this is kind of a tangent, but I think it's a really important tangent here. So again, for example, for Equilites laterofenestra, we thought we were sequencing that species across those two time points. We ended up sequencing Equilites laterofenestra and Leognathus leuciscus. And we didn't know this until we did the bioinformatics on it. And so I think this highlights an unexpected but important takeaway, which is when we're working with species we don't know much about that have very little genomic resources, expect surprises. We've uncovered many surprises in this project. Here it shouldn't be that shocking because, again, it is such a biodiverse region. There are tons of species. And especially with ponyfish, there may be prime suspects for this kind of issue because they often form mixed species assemblages, so we usually have more than one species schooling together, and they often have really conserved morphologies, which makes them really difficult to key out morphologically as well. But once we got rid of our individuals that had poor sequencing efforts and got rid of those cryptic species, we kind of looked broadly at how or what genomic changes we see through time. And so on kind of a genome-wide level, the first thing we wanted to do is see how differentiated the samples are over time. So do we see one population through time, or do we see the populations have really kind of diverged? And the contemporary individuals look very different from the historical ones. And you can see on the PCA and the admixture plots and our FST values that we don't have a lot of differentiation through time, which is what we expect. This is one population, they've got short generation times, but we generally expect to see very little differentiation. So you can see our kind of darker squares for the historical individuals and our lighter circles for the contemporary individuals overlap nicely on the PCA, they cluster together. Admixture suggested K equals 1 for both species. I'm just showing you K equals 2 plots here because otherwise it would be very boring. And our FST values temporally are very low too. So this makes sense. One population through time, checks out. When we looked at genetic diversity, I'm just showing you two ways that we calculated genetic diversity here. We see that both species lost genetic diversity. So we see a slight but significant decline in expected heterozygosity and pi or nucleotide diversity. Again, makes sense. If we expect these populations have gone through some kind of bottleneck or population decline from increasing fishing pressure or coastal habitat degradation, we would expect to see genetic diversity decline. Not super large genetic diversity decline. Heterozygosity is not really sensitive to bottlenecks, right, compared to things like allelic richness, but minor decline, significant minor decline kind of checks out. And then the last thing I'm going to show you here is we also estimated recent demographic histories for these species using MAMI-2 by fitting models of population size changes against the site frequency spectra. And we did this a bunch of different ways. And again, for both species, we're seeing kind of the same pattern. We see this deeper time historical expansion, tens to hundreds of thousands of years ago. and then a very recent and dramatic population crash, again suggesting potential evidence for a population decline or bottleneck in the intervening century. And so the brief takeaways from this section are that yes, we're seeing genetic diversity is declining in the tropics, at least in these two species. And I think I kind of glossed over this earlier, but this is a rate similar to that which we've observed elsewhere. This is at that 6% rate that I mentioned at the beginning of the talk that we're seeing in temperate taxa, in birds and fishes, and now we're seeing it in fishes, but birds and mammals and all those other species. But I think it's important to highlight we don't know how ubiquitous this pattern is. These are just two species that we've looked at, and they're very, very similar, and they were collected in the same location. So do we see the same trends in other species is something we're looking at more broadly in this project. Species that have other life history traits, maybe are more pelagic instead of near shore, have longer generation times, or not so targeted for fishing. It's important to see how all of these species are also changing to get a more comprehensive view of the trajectory of genomic change in this region. And this, again, was a bit of a tangent, but I think it's important to highlight. We didn't know we had these kind of hidden species here in our museums, and we did. And so this presence also kind of suggests a potential loss as well for cryptic speciation and reinforces the narrative that looking at diversity trends in other ways and complementary ways are also important. So looking for species loss or species turnover in these regions is also crucial. And it's important to incorporate local knowledge when you're identifying your species, because these species were only relatively acknowledged in like Western taxonomy books very recently. Okay, super briefly, I'm gonna take a little left-hand turn because I wanna highlight some work that I'm about to start that's really complementary to this stuff. So I'm a postdoc right now at Drexel in Dr. Megan Pfeiffer-Rixey's lab, and I have switched from marine to terrestrial world, and I'm looking at how, our lab is looking at how house mice are adapting to urban environments. We're basically just collecting mice from rural and urban regions and a bunch of different cities and looking for a whole host of changes. So genetic signatures of adaptation, microbiome differences, gene expression, morphology, behavior, you name it, we're trying to collect and look at it. That's all I'm gonna say about that. If you're interested, see me afterwards or go see Logan Lacey's talk or poster, who's a research tech in our lab. His poster is tomorrow. But no surprise, because I'm here in this seminar, I love temporal genomics and natural history collections, and so I wanted to see, could we add a historical perspective to this work as well, and see if genetic changes across space mirror those through time. And so recently I was awarded an NSF fellowship to extend this work temporally and see, look specifically through time in New York City and in Manhattan and see how our contemporary mice that we collected around Manhattan compare to this really excellent collection from the American Museum of Natural History that was gathered in the early 1900s. So we can see how these mice compare and how genetic changes, or what genetic changes have occurred over time. And so that's pretty much that. I think this will hopefully provide important context for any contemporary changes or patterns that we see and help us better understand the tempo, directionality, and any drivers of potential urban adaptation that we find. And that's my talk. Thanks for listening. I think I'm out of time, so I'm just gonna, one question, okay. If there's any.
I was wondering if you thought about the contribution of rare alleles in sort of this historical versus modern comparisons, because you're sampling a much, presumably a much longer time scale in historical versus contemporary population.
In terms of longer time scale, they're all the fish are collected on the same day in the same trawls, so I'm not sure I understand.
Maybe I misunderstood. In the historical collections, it's not like a long span of time.
No, it's like the fishing boat goes out, the research vessel goes out, does a trawl, might be over a few days, but yeah. Sorry if I misspoke. No, yeah. But I do think rare alleles are important and probably driving some of this. Yeah.
It's great to be here. Thank you for making it out on a Sunday afternoon. And again, thanks to the organizers for putting together a really great symposium. My name is Rob Guralnick. I am the curator of biodiversity informatics at the Florida Museum of Natural History. And if you're wondering what a curator of biodiversity informatics does, you might not be the only one. In fact, when I first arrived to the University of Florida, I said, hey, what's this job all about, right? And what I think the answer is it's really to facilitate the digitization at broad scale of natural history collections for research use and for societal benefit. So that's an awesome job and a job that I love doing. So I'm so privileged to get to do it. And really what I want to do today is talk to you about the promises and pitfalls of using natural history collections for understanding biotic response to environmental change. So what I want to do here is really cover two key themes, one of which centers around thinking about temporal dynamics of change, and especially kind of stretching that timescale back to the more distant past. And so we've heard some great talks in the session already about using ancient DNA and historical DNA. I want to continue that, but kind of stretch it back to thinking about archaeological and paleontological data and the timescales that stretch back millennia. But then I think what's really important here about thinking about temporality for me is this idea that we can also pull that timescale forward and towards shorter timescales like decadal or centennial timescales to try to link up across time to look at change happening in this sort of multi-temporal, multi-scale way, right? So that's one key theme. The other key theme I really want to touch on is this theme about thinking about the fact that natural history collections often derive different kinds of data types that we can use for inferring change. And so we know when we infer change from having use of multiple kinds of data, it provides a really nice way to kind of broaden our insights and derive new insights from integrating those different types of data. So hopefully those two themes will show up across the talk today. But before I jump into the talk itself and kind of set up a framework, I want to tell you a little bit about me. So I think sometimes we give talks and who we are and what we care about is sort of embedded in the talk but not made explicit. But I want to make it explicit because I think who I am really is driven by the kinds of questions I've been thinking about for many, many years. And part of that is certainly in global change. But I've also thought a lot about, over a long time, questions about biodiversity informatics and data sciences. And so I want to talk about both of those arenas and how I work across them. So in global change biology, I think a lot about macroevolutionary pattern and process. And so I really think about time-extended processes. My background originally was in invertebrate systematics, not in global change. And so a lot of what I think about comes from really addressing macroevolutionary questions. I also think biogeography is totally critical, right? Like we are thinking about change happening in terms of communities and lineages over time and space. There are drivers of those changes, and so much of that is really about biogeography. I also think a lot about interactions, and so I was really thrilled to see Emily's presentation today talking about interactions, multi-trophic interactions that drive systems, predator-prey dynamics that are so important, and trophic interactions that are critical for understanding ecosystem response. And because I'm interested in demographic consequences, I think a lot about population biology, right? So all those things are really critical to how I think. But a big part of what I do and think about is really about informatics and data sciences. And this has been a part of my journey as a scientist for my whole career. And this is really important work around mobilizing data, sharing it, and building community. And so what I want to see is a world where we can build communities around resources that we can all use effectively, and to kind of break down these vertical silos. And so a lot of times our data are put into repositories like GenBank or put into mobilized out to the community, but we don't always link those data well at all, right? And so building better linkages is super important. And connecting across all of this, to me, are the set of models and workflows that we can use to link up our big data approaches to our questions, right? And that revolves around thinking about new approaches like machine learning, But what I have found is that, with museum data in particular, there are pitfalls for using those data. So those pitfalls are real and they're significant. So what we want to be able to do is reduce the bias by using these new models and workflows in order to derive insights that we need for understanding change. Okay, so what I want to point out is the heart of my work is built around physical specimens. And so as a scientist, I spent my whole career working with physical specimens. And what I think is really, really critical about this is that there's so much realized and unrealized potential with these physical specimens. And critically, natural history collections provide a remarkable window into the past. And so every single specimen that we collect, every time that we collect a specimen, we can put it in a temporal context. Sometimes those contexts are deeper in time, sometimes they're more shallow in time, but every single one of those has this temporal context in which we can derive information about change that's happening that gives us that window into the past. And this is a great figure from Holmes in 2016 that I adapted a little bit to add in this longer timescale thinking about change happening over archaeological timescales, even back into the paleontological record, all the way forward into the present. And so the original figure actually just had that 1850s to present time period where we think about recent change, but I think it's so powerful to incorporate the archaeological and paleontological record as well. So what I want to do in this talk is really provide a framework where we can look at this multi-scale temporality and basically use that temporality to derive insights about global change. And I'm going to do that using a set of vignettes that are going to be at a high level, kind of driving home some key messages. And I hope that these vignettes show the power and some of the limitations of a data-intensive approach and how we can connect across data sciences and biodiversity, okay? So I'm going to start with thinking about deeper time perspectives and global change and human impact. And the example I'm going to use are parrots across the Caribbean islands. And there is a beautiful record of parrot samples and specimens across the Caribbean. These have been well collected over many, many, many years. It's a record that extends from the paleontological side to the archaeological side to the modern side, and we have great data. We can't always ID the specimens we're looking at, but we have this remarkable record collected across islands. And so in a paper from 2023 led by Jess Oswald, a former postdoc, and a set of collaborators, including Julie Allen at Virginia Tech, Michelle Lefebvre, an archaeologist at my museum, Dave Stedman, and Brian Smith, we were able to basically create a baseline of biodiversity using radiocarbon dating and ancient DNA of these specimens. So critically, this is a baseline before human impact. And during human impact, as Amerindian peoples began to settle on these islands. So I'm going to not give you the whole story about the work that Jess did. What I want to do is focus in on a particular species. This is Amazon ventralis. This is a Hispaniolan parrot. The Hispaniolan parrot is currently found in the Dominican Republic and only there, okay, on one island. But looking at the paleontological record of ventralis using radiocarbon dating and ancient DNA, we're able to reconstruct a much broader distribution in the past. This parrot was found across the Bahamas in the paleo record and was found in the archaeological record on Montserrat and on the Turks and Caicos. Now, importantly here, and what I want to point out, is that we don't have archaeological evidence for the Hispaniolan parrot on the Bahamas, and we don't have paleontological records of that species in Montserrat or the Grand Turk Island. So what we can infer from this potentially is that this parrot actually was around through the Holocene. When humans arrived into the islands about 1,000 to 2,000 years ago, we think this led to the extinction in the Bahamas, the extirpation of those populations in the Bahamas through human arrival. So they're in the paleo record, they're not in the archaeological record. Probably this is a human-caused extinction. Okay, but what about Montserrat and the Turks and Caicos? Well, let me show you the distribution of those islands in case you're not familiar with Caribbean biogeography. So Montserrat's pretty far south from the DR or the Bahamas. And of course, it's possible that the population of the Turks and Caicos archaeologically might actually be a native population. But in Montserrat in particular, we find this in the archaeological record, but we don't find it in the paleo record. And I think that might be a pretty good indication that this was a translocation of the population from presumably the Hispaniola area into Montserrat. So basically, humans were moving these parrots. They were causing their extinctions for many, many, many generations before we ever get Europeans settling into the Caribbean. And of course, the Montserrat population also goes extinct on European arrival, and so it's no longer there. So now we have this better context for looking at the distribution of parrots over time through the lens of natural history collections, where we're using radiocarbon dating and ancient DNA. So I'm going to move from the balmy waters of the Caribbean to the much cooler conditions up in the Arctic. And we're going to kind of scale back in time a little more to the last glacial maximum. And then we're going to move forward in time to forecast into the future. And we're going to use an example here from collared lemmings. This is our Vickling rodent. This is work from Stefan Prost in 2013 that I was involved in, where we use fossils and modern records, including ancient DNA and modern DNA, and distribution modeling to tell the story about the distribution and demography of mammals in the Arctic. And for this one species, I just want to point out, we can use the fossil data and the modern data to kind of calibrate our models and constrain some of our commission errors to get the predicted distributions in the last glacial maximum about 18,000 years ago in the present, and then we can forecast into the future, which is tricky and hard, but under carbon emission scenarios, we can basically make forecasts about where the species might be into the future. And what we can see is, the species was more widespread in the last glacial maximum, and its population numbers are certainly higher based on inferences using Bayesian approaches. And look at the skyline plot here on the bottom part of the figure. We can see that populations dropped in the Holocene and didn't recover. And certainly in the present, and actually clearly in the future, we're losing habitat, and we'll continue to lose habitat and continue to have more disjunctions in these populations into the future. So I think that's useful. We can begin to talk about change dynamics over time in terms of distributions. But I'd argue niche models aren't really the best way to address some of these questions. If we're talking about conservation outcomes, 50 or 60 years in the future, it's too late for a conservation outcome probably. And we could argue that what we really want to do is look at decadal timescale questions about change. And so how can we kind of tie it together across these timescales? That's what I want to turn to next. So I'm going to shift gears a little bit more again. I'm going to stay in the same region, but I'm going to move to thinking about Arctic butterflies. I'm kind of wandering around this whole area of different groups, right? We've got some mammals, birds, butterflies, all shaken out. But what I want to do here is talk about changes over shorter timescales, a 50-year time period. And this is work with Von Shirey, who was a graduate student of Leslie Reith. This work was done in Leslie's lab when they were a PhD student. They are currently a Smith Fellow postdoc with me and Melissa Guzman at USC. And in the Arctic over the last 50 years, you can see a strong signal of warming across most of the Arctic. And you can see as strong a signal as 4 degrees Celsius just in the last 50 years in terms of climate warming. We also see a pretty good record of butterfly occurrences and preserved specimens across the Arctic region, Boreal Arctic region, much more densely sampled in the south. No surprise, in the north, we see a lot less sampling because no one lives up there to sample butterflies. It's very hard to do the work up there, and there's literally no one there to do the work, or very few people there. And we have a pretty good record over time, but it's a pretty sparse record. It's not that well sampled. And Oliver, here's your declines in the present for sampling for butterflies as well, right, from the past. So how can we look at change in terms of butterfly occupancy or distribution over time, over this 50-year period? It's really tricky. And the problem is that museum data are incidentally collected. And because of that, it's very challenging to basically fit the observation process. We don't have covariates to fit it often, and often our data are highly biased. They're highly biased in space, they're highly biased in time, and they're often highly biased in both dimensions at the same time. So how do we pick up that signal? So let me just walk you through this conceptual figure really quickly here. What we're really interested in is the ecological process on the top left. Okay, that's the signal of response that might be occupancy change or abundance change in relationship to climate change, and that's a signal we're trying to reconstruct. But without those covariates to fit the observation biases, and without structured sampling where we can fit that in our sampling method, we might miss that signal. And that's what you see on the bottom right. And so the true process we're trying to model is that gray line. The inferred process that we model is actually off that gray line. That's a dark black line. And that can be off by quite a bit. We can miss the real signal quite a bit without accounting for those biases. So what do we do? So I'm going to show you a method that Vaughn developed that kind of tries to account for these challenges about bias using basically an approach based on community sampling. And so what Vaughn developed is a methodology where they can take all those observations I showed you in the Arctic. They basically grid up the Arctic into 100 grid cell pixels or regions, and then they basically create 5-year time windows within each region over the 50 years. So a total of 10 blocks, right? 10 time blocks over the 50 years. And that's step one. So basically step one is do that assembly of the regions, the time intervals, and for each observation, we basically assign those observations into that region and the time window where it belongs, okay? Step one. Step two is where the magic happens. This is really imputing detections on detections. And the key here is subdividing that 5-year time block into 1-year visit intervals. And this is important for us to be able to basically separate the detection and occupancy process properly. So we create these visit windows, and if 2 species are found within that visit window, and 2 or more, we can basically assume that we can account for non-detections properly. It turns out, based on simulations and a whole bunch of data and empirical work, that assumption of having a very sparse set of information about community sampling is enough to reduce the bias. It doesn't completely remove the bias, and it creates high uncertainties. But even that limited amount of information is enough for us to basically filter the data that conform to that filtering, and then move into downstream modeling. So Vaughn was able to basically fit occupancy models, look at occupancy shift rates over time, and what they did next is to basically ask some very simple questions. How does occupancy shift vary according to species traits? And the traits that were of interest were niche breadth, mean temperature niche, and body size and phylogeny. And so they fit these models. And by far and away, the models that were best supported always included mean temperature niche, okay? We get a couple of cases where models are nearly equivalent for other kinds of variables, but in all cases, that mean temperature niche is a really important signal. And to our surprise, we didn't find niche breadth as an important factor, nor is phylogeny a strong factor or covariate in these models either when we look at phylogenetic autocorrelation. And so on the figure on the left, what I'm showing you here, I think is sort of the key figure about what we actually found. This is looking at basically shift rates to higher occupancy, towards more occupancy, or reduced occupancy. And the warmer adapted species are the ones that are basically increasing their occupancy in the Arctic. And that's no surprise, right? Those are the species that are probably in the boreal or temperate regions that are moving up into the Arctic, and as they expand upwards, their occupancy is increasing. The cold adapted species towards the bottom of that figure, on the left-hand side of that figure, those are the ones that are decreasing in occupancy, and those are the ones that are basically losing habitat as climate warms, even over that 50-year period, right? So now we're able to kind of scale across those timescales between, say, millennial timescales and decadal timescales. And I think importantly here, I want to mention this to be clear, this also has really important outcomes for conservation. This is a timescale when we can look at species that have the most declines over the last 50 years, and those are ones we can really focus on to basically make conservation decisions and do conservation prioritization. And that gives us a quantified measure of that rate change, right, that we can use to help us. Okay, I have one more vignette, and then I will wrap up here. And this is a story about mammal body size and urbanization. Any mammalogists in the room by any chance? Anyone? Cool, we have a couple mammalogists, great. Every single rat catcher in the audience knows. And when you go out and you field trap a mammal, you do a bunch of external measurements, right? You measure body size, you measure some tail length and total length, and those all show up on labels on your specimen. So this is a pica taxon I absolutely love and have fond memories of in Boulder. There's a label on the pica that shows measurements, as well as a whole bunch of other critical information. But on the bottom of that label are a set of shorthand measurements about the external measurements of that specimen, right? And those are mobilized into repositories. So there's lots of data mobilized about traits that are stuck in digitized specimen records, and they have not been unlocked. So what we were interested in doing is unlocking those trait measurements from digitized records. And so what we did was we basically took approximately 30 million digitized vertebrate records. We decomposed those records to extract these hidden traits, and we harmonized the outputs. And the big deal here, if you're a mammologist who's seen these abbreviations, it's not standardized across collections. So one collection can put it one way, and one collection can put it a different way. And so harmonizing those data, creating the right units around them, and then basically reassembling them into data that can be used for research is a real challenge. We wanted to make sure we could do that and serve that to the community with the fairest possible use, findable, accessible, integratable, and reusable, and make it reproducible as well. And so we use natural language processing to basically do that task. And we were able to extract a lot of data, like a lot of data. So 500,000 mass measurements for mammals alone, right? For many species, about 400 or so, we have 100 to 1,000 measurements, and for a few species, we have tens of thousands of measurements, right? That's pretty cool. And it's not just mass, there's all the external measurements, there's reproductive status data, embryo counts, all kinds of really valuable information about what's going on with individuals in populations across a wide range of taxa. So we look at a map, we can plot those. Those data are biased spatially, they're biased temporally. That's not a surprise, but there's a lot of data to use. So we had a very simple set of questions. How does body size vary with climate, and in particular with mean annual temperature and urbanization, just across space, okay? And also critically, we're thinking about traits. So we're thinking about nocturnal versus diurnal mammals. We're thinking about hibernators versus non-hibernators, big versus small. So the first result should come as no surprise. There is a relationship between mean annual temperature and head body length for both small and large mammals, just what we expect. This is Bergman's rule for mammals, and mostly makes sense. We expect that there are bigger individuals in colder environments within and across species. And that turns out to be true. But what we did not expect at all is this really strong urbanization size relationship. And that relationship is positive. It means that as you go into more urbanized environments, mammals tend to be larger. The higher, greater head body lengths, and it's true for mass. That we did not expect. So we also looked at it over time. So we fit models across a single species, in this case, Paramyscus maniculatus. This is the deer mouse. That is a very cute mouse up there on the screen. And we basically were able to kind of locate areas where we had enough sampling over decades to begin to look at the signal over time in those rectangular regions. So we looked at that change rate in body size in relationship to the change rate in climate and urbanization. And we do also find that deer mice are getting larger over time and over urbanization gradients. So why? We think this probably has to do with food resource allocation across urban environments compared to non-urban environments, but it's a very strong signal, surprising signal, and there's definitely more work that needs to be done. Some species are going to be more cryptic in urban environments, or there might be a variety of responses that we're not picking up here by just looking at these simple analyses to start, but more to do. And then finally, we made these data all available, and we kind of broadened out what we're able to serve out across different traits, and really linking across archaeology and paleontology as well to create kind of a global trait resource for sharing individual traits from specimens and other resources called Futurists. And I encourage you to check it out. It's a cool project. You can go to futurists.org and learn more about it. So I am done. I have a couple thoughts to impart on my way out and conclusions. Basically, what I wanted to convince you of is that natural history collections can provide this critical baseline data to understand change, and not just in the recent past, but farther back in time at millennial and even longer time scales where humans have had impact. And what we know now is that human impact has been happening for thousands, tens of thousands, and potentially millions of years. We've always impacted the environment. So our collections provide this critical resource for understanding those longer term baselines. I think these are hard to use properly still. So even though we can do this kind of work of looking at multi-temporal dynamics, there are challenges using these data, and new models and workflows provide a really nice opportunity for us to begin to basically winnow down those biases and get the signal from the noise. And I think that's really exciting. And I think it's particularly exciting because we're entering a different world in the next decade than I ever lived in as a scientist. I think with machine learning tools and other approaches that we're developing, we're going to be able to scale up and automate a lot of these tasks more efficiently and effectively. It's just a really exciting time to be doing global change biology with natural history collections data. So thank you so much for your attention. I'd be happy to take a few questions, please. Thank you so much. Yes.
Yeah, so I'm very interested in that urban aspect. Do you have data other than mammals that you've been digitizing for these things? I know response to urbanization really seems like dependent on the animal you're looking at.
Totally, and the traits, right?
Right.
And yeah, we tried really hard in this paper to address these questions about interactions between traits and environment, including climate and urbanization, and those interactions are really critical. We do have data for birds, herps, fish. Fish are probably not as relevant for your urbanization part. Yeah, we're busily trying to get those data assembled. We're really careful about making sure those data pass a bunch of pretty significant quality checks. We do a lot of work to build tools for outlier detection. There are issues about, is this a juvenile or an adult, that we've had to really struggle with in terms of the distributions of juvenile adult body sizes. So when I say that we kind of didn't, like we had a recipe and like we got all this data extracted, it sounds awesome, but I can tell you is truly painstaking work to clean up those data to the point where we think they're usable. Yeah. So they're coming if you're interested.
Yeah, very.
Awesome. Great. Yeah, any other quick questions, you guys? I'm not going to leave the stage until my time is completely allotted. So, Corey, yeah, what's up?
Could you look at whether competition was driving some of body size or if it's like co-occurring mammals?
I love that idea. That's a great idea. Sure you can. I think we basically have barely scratched the surface of what we can say here. Is that my time? Am I actually now officially out? 1 minute. 1 minute. Is that good enough for now? Okay. Yeah, I mean, I think the point of those papers is more like almost exploratory with some very light hypothesis testing, like, we think urbanization matters, does it really or not? And so we basically feel like there's so much more to do here. I know people at this meeting have a lot of interest in urbanization and evolutionary and eco-processes around urbanization, right? One more question real fast? Yeah. Oh man, so that's $1 million question. We should talk about that separately over coffee. I have ideas. It comes down to capacity. It comes down to building community and having communities really focused on the task and knowing how to get to where they want to go. And it's not probably because people don't want to do it. I think there's just still, after all this investment in digitization, there's just still more to do.